Zhang. He got his PhD in drug design and medicinal chemistry from Shanghai Institute of Materia Medica, and then he did a postdoc at the University of Colorado at Boulder, studying cancer and immunology. After his first postdoc, he did another postdoc at the Scripps Research Institute, studying DNA damage, metabolism, and aging, before becoming a research assistant professor and drug discovery core manager at the University of Minnesota. Now he is assistant professor here at the UGA in the Department of College and Pharmacy, working on elucidating drug mechanisms and identifying actionable targets and biomarkers across aging and disease models. Today, he will discuss some of his work in his talk, Bioinformatics and Machine Learning and Aging Biology and Drug Discovery. Let's welcome him.
Thank you, Kevin. Thank you for the opportunity to be here. I think I successfully get someone's attention about aging, the importance of aging, no matter what gender you are, what race you are, what political views you are, we all age. Aging comes for every one of us. So in my lab, we want to tackle the aging by drug discovery. Hopefully we can develop anti-aging drugs, or we call therapeutics, to slow down the aging process or even reverse it. So today, I want to focus on the bioinformatics and the machine learning and how we can leverage this technology in both aging biology and aging drug discovery. So first question, how do I get here? Why I'm studying aging? Actually, you can see my background was in medicinal chemistry. I was used to work on type 2 diabetes. I will show you the story later. So this has to trace back to the nightmare when I had when I was young. So I was always thinking about the vastness of the universe and the tiniest of this planet and the significance of this life. So this makes me so terrified of death, especially when I was doing my first postdoc in Colorado. So because I see the past of my grandma, so that the death haunting me like every night. Until someday when I was reading or watching a TED Talk. This guy said the first human being who can live to 1,000 years have been born. So that was so shocking and mind-blowing to me. So is that real? And I did some research with a scientist. I want to know, is it hype or is it real? And now it's real. And these two monkeys and this one human changed my career path totally. So you can see these two monkeys are actually at the same age. The one on the left is on caloric restriction. 70% of the diet for 27 years. The right monkey can eat how much he wants. Then turn out they look like not brothers, but father and son. It's shocking that we can just tweak the diet and change the aging process. So then I decided to do something with this. So I was working on cancer. So at the beginning, you can see I was very young when I was in graduate school. I was studying medicinal chemistry, working on type 2 diabetes. I also work on organic synthesis to synthesize the drugs I designed. And at that moment, my dream was to cure diabetes. But to do that, I need to learn more biology. So when I did my first postdoc in Colorado, so I expanded to biology, biochemistry, a high school drug screening. The topic I was studying was cancer. The more I learned about cancer, the more let's say pessimistic I became. I don't think we can cure cancer because it's so complicated than we thought. Because the one human and the two monkeys, I changed my career path. I was ready to do a faculty, start my independent career when I was in Colorado. But I don't want to study something I have no confidence for the rest of my life. I was so fascinated with aging, so I did, actually the story is more complicated than this. I even tried to get a second PhD in gene editing and stem cell. So I really did that. But turned out I was recommended to go through a second postdoc if I really want to do this. So I went to Scripps Research Institute, and using my expertise in drug discovery, I'm working in Dr. Paul Robbins' lab, who's the big name in aging field. Then I just work like crazy. I want to learn anything that I can, aging biology, in essence, high content imaging, multi-omics, and recently machine learning, bioinformatics. So here I am. So studying aging, which is my nightmare and also my passion. And looking forward to future, the AI shows me I will look like this when I become 80. So that was real nightmare. So what can we do? What can I do? So can my research to help me reverse, slow this down a little bit, not only for myself, but for everyone. So that's my career path in a simple slide. But the aging is not like an individual problem. It's not my problem. Actually, it's a global problem. It's a huge challenge. So you can see the whole world is aging. Actually, just in 10 years, we'll have more elderly people than children. So you can imagine this is not only impact the population and ratio, but also economic, the health care burden, everything. So obviously, studying aging is not only important to individuals, but most importantly to the whole society and humanity. And scientifically, aging is the driver of most chronic diseases, right? If not all. And you can see that with increase with age, all the diseases, especially chronic disease, increase exponentially with age. And in my lab, we view aging is not just a natural process. We think aging is actually the root cause of many diseases. And turns out it's true. So we are developing drugs targeting the root cause, which is aging, and use the same drugs. We can treat cancer, we can treat skin diseases, diabetes, many others. So we are currently working on cancer, skin issues, and neurodegeneration with one same drug. So which means we don't need to tackle this disease individually. We don't see them as separate problems. Actually, they're interconnected at the bottom. So is that real target? How can we target aging, right? Where do we start? So thanks to the advances in the past several decades, we have identified the several we call hallmarks of aging, the mechanism that can drive aging process. As you can see, the hallmarks of aging increased from 9 to 12. I believe there'll be more mechanisms of aging will be discovered in the following years. But the beauty of this, that aging is malleable. That means you can tweak it, you can slow this, you can reverse it by genetic, by a diet, or by exercise, right? Or you can do that by drug discovery. That's what we are doing in the lab. My cats, 2 cats, they are trying to slow down their aging process. So hopefully I can develop drugs for them very soon. So this is the overview of my research. So we are leveraging almost everything that we can to develop gerotherapeutics or anti-aging drugs. So we can do drug screening, either virtual screening or high-throughput screening, high content, imaging-based drug screening, or we can design drugs, use traditional medicinal chemistry methods or CEDD, or we can leverage AI and machine learning. Then once we get a drug, then we can test the therapeutic effect in aging models for lifespan and health span. If they work, then we can move on to individual disease because we are targeting the root cause. Now we want to see drugs improve individual diseases. So now we have about 4 drugs in the pipeline. So we're testing this in cancer models and neurodegeneration models and fibrosis models. Hopefully they will work. So we just get started, but we are very optimistic about these drugs. So once we know the drugs are working, so the next big question is, how do they work, right? Aging is so complicated. How do we know the drug target of aging? It's so challenging. That's why we will need to use multi-omic studies, bioinformatics analysis, machine learning to help us to dissect the complex biology in aging and disease. Then once we figure out a new target or new biomarkers, we can go back and develop new assays to go back to traditional target-based drug discovery. So that's kind of the overview of research about that. Today, I want to emphasize that bioinformatics and machine learning play important role in the whole process. If you look at the biology, we can see this at different layers. So biology can be seen from genetic level, transcription level, or translational level, metabolite level. And thanks to the technology, that we can generate high amount of data. Then with this data, we can look at the biology from different layers, and we'll generate unlimited amount of data and a lot of data sets. So how do we analyze these data sets? So data doesn't mean knowledge. So it can be noise. So we have to leverage statistics, algorithms, machine learning to help us to find the trend, the pattern, then get us discoveries or decisions. For example, from this, we can get new biomarkers, drug targets, and also use this technology to help us design better drugs. And eventually, we can turn this data into interventions. That's my view on how important bioinformatics and machine learning, all of these computational technologies to aging biology and drug discovery. So you can type in bioinformatics in any biology field, like drug discovery, aging, you can see this is the future. So with the increased amount of data publications and attention. So I believe we are in a very promising area that we may be able to tackle complex diseases like aging. So in my lab, well, in the long term, we hope to tackle every hallmark of aging. We can't do that all at a time. So we are planning this in different tiers. So currently we are focusing on cellular senescence, DNA damage, chronic inflammation, and now we're also get into the second tier to look at epigenetic modulators. In the long term, hopefully, we can develop drugs that can target every hallmark of aging. But today, I want to show you use cellular senescence as a case to see how bioinformatics and machine learning can play a role here. So what is senescence? Just to make it simple, so we have cells in our body every day, and cells are exposed to stress, a lot of kind of stress, right? UV light, oxidative stress, DNA replication errors. All these stress can cause cells to make a decision. Should I stop dividing, or should I pass it on the damage to daughter cells? Right. So this limbo status, which we call senescence. So cells, they don't die and they don't divide, but they metabolize. They release these factors, inflammatory factors, so they can help. At the beginning, they can help tissue repair. They can help development. But if they accumulate in the tissue for too long, they will drive all this chronic disease, for example, cancer, diabetes, neurodegeneration, many kinds of diseases. So senescence become a very promising low-hanging fruit for drug discovery, aging drug discovery field. As you can see, there's so many papers have shown that with age, we have accumulate senescent cells in almost all tissues. And if you transplant senescent cells, or we can zombie cells, into healthy organisms like mice, you can make them age faster. And on the contrary, if you remove them, like through genetic approach, remove the zombie cells, mice live longer and healthier. So this is just not only the observation, it's the causality of senescence. So that makes targeting senescent cells very exciting field right now. So there are two approaches we can develop drugs to target senescent cells. One kind of drug we call senolytics, the drugs that can kill these senescent cells. Or we can develop drugs that can suppress the status of the senescent cells without killing them. For example, we have senescent cell in the brain, right? Your neurons may get older. You don't want to kill the neurons, but you may want to suppress it. So that's why we want different kind of drugs that can target senescent cells. So this is just an illustration we just submitted to the cover art of the journal. You can see we can call temporarily call these senescent cells zombie cells. Right? They don't die, but they're contagious. They can release these bad signals and turn surrounding healthy cells into senescent cells. That's why this is a very good analogy calling senescent cells zombie cells. So before introducing my work, I want to ask you guys, if you were given the task, how would you develop the first senolytics, the drugs to kill senescent cells? So the first senolytic was discovered 10 years ago. It's a very new field. So how would you start? So first, usually we need a drug target, but at that moment, we don't know much about senescence, and there's not many drug targets we can do. So here, bioinformatics, transcriptomics play a significant role to the discovery of the first senolytics. So you can make cells in two groups. You can make healthy cells and senescent cells and sequence their transcripts to see what genes that keep these senescent cells alive and what's the difference between healthy and unhealthy cells. So they find out these senescent cells are zombie cells. They have upregulated anti-death programs. So these cells are not just staying alive. They are actively resisting death. So that means we might leverage this program to develop drugs. They screen the 20,000 genes and get narrowed down to 6 potential transcripts as drug targets. And they validate by PCR and Western at both at transcription level and translation level. Then they map these genes to a network. You can see this, what they call a SCAP, senescent cell surviving pathways. So these zombie cells rely on these key gene nodes to fight death. So how can we leverage this? So they pick existing drugs that can target these nodes, then combine these drugs to target most of these SCAP, senescent surviving pathways, so that we can kill them cells, which was genius. Yeah, that leads to the first senolytic combination, DNQ, dasatinib, and quercetinib. And they show this in the mice. So DQ can kill these zombie cells in different tissues, like the fat. Usually, we have a lot of zombie cells in the fat. And not only reduce the cells, but also improve the function of muscles and the health span. We call composite aging score. You can see tremor, kyphosis, all this phenotype, functional phenotype has been improved. You look at the mice, they were cleared from these zombie cells. They look healthier, they look more energetic, more functional than these untreated cells. So that further value that senolytic is promising. So far, DQ has been on different clinical trials. That's why I say the beauty of targeting aging. You have one drug that can target the root cause of aging. You can apply this to many diseases. So you can see DQ has been applied to so many diseases and now in different clinical trials for different conditions. So this is the first successful discovery of senolytics. So far, we still have, we don't have enough senolytics. We have a few senolytic and cystenomorphic from natural products or repurposed drugs, but the challenge are still significant because senescent cells are not the same. Some drugs work to certain senescent cells, but doesn't mean they will work to others. We need more senolytics that can kill all kinds of senescent cells, or we can call pan-senolytics. Also, these drugs have different side effects or specific issues or bioavailability issues. So definitely we need more senolytics so that we can eventually move on into clinic and to treat real patients. So how we can do to develop the next generation therapeutics. So you look at the traditional drug discovery approach. First, you need a target. So besides the BCL2, that's kind of like not very perfect drug target, but that's the best target we have so far for senescent cells. So you need a drug target, then develop assay, then you do all the drug optimization to clinical, preclinical, and clinical trials. But given the challenge or heterogeneity of senescent cells, this traditional drug discovery approach is not working well. So what we think, maybe we can do a different way. How about we develop an assay to look at the phenotype of some cells? Then we throw drugs on these cells. Just look at the phenotype. Can these drugs kill these zombie cells? We don't care about the mechanism. We can't decide the mechanism yet. Then if they work, then we will go back, use all kinds of techniques, like especially bioinformatics, multi-omics, and machine learning AI to go back and study the mechanism. That's what we did. How do we get this assay? So we had to develop a phenotypic assay. So senescent cells is characterized by the high elevation of acid-beta-gal senescence associated with beta-galactosidase. So we can develop a small molecule, a fluorogenic. So you give this small molecule to the senescent cells because they have high level acid-beta-gal. You will cleave the drug and make it senescent or green. So the greener the cells are, the more senescent. So now we have a phenotype assay. So with this, I established this senescent therapeutic drug discovery platform years ago. So we can induce any kind of cells into senescence, then screen any kind of drug libraries. Then in this way, we can develop either senolytics or xenomorphics. If drug candidates or hits are good enough, we can move directly into mouse studies. If not, now come to my graduate school work. We can do traditional medicinal chemistry optimization, design the drug into better version and do several rounds of drug optimization until we get an optimal drug lead. Then we move on to preclinical and clinical. So this is kind of like a perfect platform that we can develop next generation therapeutics. So with this platform, I have been discovered so many classes of senolytic and xenomorphics. You can see this here, including fatty acids I'm going to talk about today, flavonoids, FDA repurposed drugs, or kinase inhibitors, all these. And another example I want to share with you is machine learning. How we can use machine learning to predict senolytic or therapeutics. So these drugs have been validated in cell culture, in ex vivo tissues, and in vivo different mouse models for health span and tissue analysis, and show they truly impact many tissues. So first, I want to give you a flavor of this drug discovery story. So at the beginning, we picked this fatty acid library. So it's intentionally created. So don't worry about this chemistry. So the goal is not to put random drugs together. We want to know through the screening, use drugs do not work. We want to know why they don't work. What structures lead to that? If they work, why they work? Right. So this is very intentional. We call structure-activity relationship. So we built this kind of library that incorporate different chain lengths, saturations, configurations, and metabolites. Very complicated. It's chemistry. And long story short, we identify a special class of fatty acid, or ESA. So they are very promising in this preliminary screening. And then we expand this to different cell types. As we said, some senescent cell type, right? They are so heterogeneous. Drugs working in one cell type doesn't mean they will work to other cells. Then we test in different senescent cell models. You can see alpha ESAs that work across all different kinds of senescent cells, which is very promising and very, very selective to non-senescent cells. So they can selectively kill senescent cells, not a general toxic drug. You look at the images, you can see green cells are zombie cells. You put these two drugs in these cells, they can kill the green zombie cells at this concentration. We do dose response. You can see higher dose, you can remove almost all zombie cells. Then we did mouse study, right? Do they work in vivo? So we do short-term treatment because zombie cells, usually they just need 1 week to build up in your body. So we treat these mice just for a short period. You can see they can reduce the senescent burden in different tissues. And if you treat the mice in long term, you not only reduce the tissue senescent cell burden, but also improve the function and the phenotype. So especially tremor kyphosis, you can see the mice treated with the ESA, they have lower composite aging score, meaning they're healthier and more functional. So you also need to look at histology to look at more evidence. Can these drugs reduce the senescence in these different tissues? Yeah, which the not is true. So now, finally, come to the next big question: how do they work? Right. So this is about cell death. So intuitively, we want to look at what kind of cell death program was triggered by the ESA. So we use different inhibitors of the cell death. So if the inhibitors can rescue the cells from death, that means these drugs will trigger the apoptosis larva. So turn out these drugs did not induce death through apoptosis or natural apoptosis, but the phagocytosis, which was new at that moment, because most of the senolytics discovered so far are targeting apoptosis. So then what is phagocytosis? I'm not going to get into this. Phagocytosis is basically a very complicated process. Just to make it simple, it's an iron-dependent lipid peroxidation. So the key two factors depend on lipid and lipid peroxidation and iron metabolism. So we use inhibitors of these two components and find out we can rescue the cell death from this. We further validate that phagocytosis is the death mechanism. This is, we further stain the cells. You can see this is zombie cells that have more ROS or oxidative stress, and they have more iron levels. That makes them accumulate more lipid peroxidation. So these are all phenotypic level. So of course we want to dive deeper, but before that we want to see how the chemical structures of the drugs cause this phagocytosis. So I know there's another country audience, so I'll skip most of that. So basically, what makes these drugs unique or different than the other fatty acids is that they have the conjugation and double bond system, make this more stable if they are being oxidized by these free radicals. So that's why these ESA drugs, they work like a fuel. So senescent cell or zombie cell, they have a lot of ROS or oxidative stress. You add fuel to the fire and make the fire bigger, so cells will get pushed to the limit and die. So that's kind of mechanism. Then to validate this chemistry hypothesis, we test more fatty acids and turn out it's true. With only fatty acid with this conjugation system, they can cause senescent cell death. If you break this conjugation system, they don't work. Right? Very beautiful. Then we have a chemistry hypothesis. So senescent cells have this higher iron, higher loss is already pretty dangerous, then ESA is kind of field, add fuel to the fire, then cause cell death. So this is from the chemistry. Of course, we want to know, are there any other mechanisms? How can we do that? You have to go back to the bioinformatics. So we have to study this in an unbiased way. So we have to look at all the signaling pathways, all the genes. So before we carry out any wet lab experiment, we use computational prediction. We tell computer the structure of the drugs and let the AI to predict the possible interaction pathways. You can see computer telling us these two drugs, ESA, and mesothelin, in a high probability of targeting apoptosis. That further validate our hypothesis is through apoptosis other than apoptosis as shown in other drugs. Then we map these signaling pathways together. You can see these drugs are mostly disturb the fatty acid metabolism and the apoptosis signaling pathways. And they are highly overlapped through these regulators in this direction. Then we run real wet lab experiment. So we collect cell samples, treated, untreated senescent cells, healthy cells, then run the whole transcriptomics analysis, then map the signaling pathways. As you can see, the ESA, the drug, can increase this fatty acid metabolism and fructose regulate pathways. You block this pathway, you will suppress this. You block this pathway with inhibitors, you will suppress all this signal. So then you look at the top genes that related to this apoptosis. You can see that antioxidant genes are downregulated, and on the opposite, the pro-apoptosis genes has been upregulated. And we also validate this by PCR to look at individual top genes. Now, the top genes shown here are including ALOX15, ACSL4, and LabCat3. So they are at different arms in the fructose signaling pathway. So these genes are in the lipid metabolism pathway, and others are in the antioxidant arm. Then we dock these proteins with a small molecule with computational methods. So you can see that these drugs are more likely to bind to these lipid remodeling targets other than the antioxidant targets. And so then we validate this in the wet lab by inhibiting these three proteins. You can see we can totally rescue the cell death from that, further validating these three playing important role in this ESA-induced apoptosis program. Then zooming into the top gene, which is ALOX15, we want to know how, at a molecular level, how the small molecules bind to the protein. You can see that the ESA take a very special L shape and bind to the allosteric site of the protein. And the other kind of acid, like the substrate acid, arachidonic acid, taking like a U shape. That makes them make this ESA particularly good binder for the allosteric pocket. So then we further validate this through functional assays. So this ESA can increase, not only bind to the ALOX15, but also increase its functional activity, particularly enzymatic activity. So that further confirm these guys are the target. The last step, we want to be very sure, so we run target lipidomics study. At all the lipid, because the ESA is a lipid and it's targeting the lipid remodeling program. And you can see the lipidomics data also points that this ESA has been integrated into the cell membrane. That will cause the cell membrane damage, eventually cell death. So based on all these data, computational bioinformatics, analysis of network pharmacology, then we've identified a biological model of cell death. So first, ESA can be turned into oxidized form, which is dangerous to cells. Then this oxidized lipid can be integrated into the cell membrane through these three proteins, ALOX15, ACLCL4, and LAPCAT3. So these oxidized lipids get into the cell membrane and cause membrane damage, eventually lead to cell death. So now we have like 4 picture of how these drugs work to kill zombie cells. Because zombie cells have this, they are prone to ferroptosis because they have high levels of iron and ROS. Also, they have higher levels of lipid remodeling program. And the ESA is leveraging this vulnerability and push them to limit, then cause lipid ROS and eventually cell death. So that's kind of the mechanism. So from this, you can see we identified the first in class lipid synthetics. And we also identified new mechanism. Unlike traditional synthetics that targeting apoptosis of necroptosis, these ESAs are targeting ferroptosis, which is new. And to study the mechanism, I can see we need almost everything. Right? You can't just pick a protein and study that. You have to look at the whole different layers in the biology in an unbiased way. That's why we have to use multi-omics studies, use AI to dissect this so complicated network and the genes in the cells so that we can unbiasedly identify the mechanism of drugs. So as you can see, through this phenotype-based strategy, we can target complex diseases, particularly aging or senescence. Then you will have the drug candidates. We can leverage bioinformatics, machine learning AI to dissect the mechanism, to identify the mechanism or new drug targets or develop new assays. Lastly, if you are interested, because this ESA of natural products, they're from mostly from bitter melon. So that may means it's more bitter melon. You can clear the zombie cells in your body. So lastly, you have time, I want to talk about machine learning, how we can leverage machine learning in aging biology and drug discovery. So I want to touch on that very briefly. So from our other projects in the lab, we have built a very unique in-house data set, active drugs, inactive drugs with different chemical structures. So traditionally, we use human experts to design next round of drugs. But we are curious, if we feed the data into a machine, can AI or machine learning algorithms help us dissect why these drugs don't work, why those drugs work? So we turn this chemical structure into a language that machine can read, like through smiles. Then we train the machine through both positive and negative data sets, then through different algorithms, and we pick the top ones. Then use this trained model to test or predict more active drugs. So we pick this coconut natural product library, and turn out, when I look at the predicted hits, most of the top hits don't look like drugs to me. So so the prediction is not that ideal. So then I pick some of the some hits in the middle that machine cannot the algorithm cannot make them on the top. And that's based on my human institution. I bought these drugs and I test them, turned out my guess is right. Then I feed this data into machine learning algorithm and retrain the algorithm. So it's kind of a biased training or reinforced learning process. Then we got these top genes top drug candidates look like more drug candidates. Then we test them in cell culture, and turn out it's true. They are very promising new drug candidates. And you look at this top drug candidates in the chemical space, they are clustered together. That means the prediction is more reliable than the first round of training. Then we are currently in the last step of the small studies to validate this new drug hits identified from the machine learning and the human domain expert collaboration. So in the future, this is the first attempt to work with machine learning through human domain expert. So in the future, we're going to try to optimize every step of this pipeline to make the models better and more accurate and more predictable. So, and even more generative, not only predict existing drugs. So in the future, we want machine or AI can generate the better drugs for us, so we can synthesize them, the non-existing chemical structures. So that's a future direction. So last slide, as you can see, either in biology or drug discovery. So it's a very long, expensive process. It takes 10 years, billions of dollars, and the success rate is very low. But with the technology of bioinformatics, multi-omics, and especially AI and machine learning, and we believe the drug discovery and development process can be improved in the following decades. And but still, we need to work together with human expertise, and only combine these forces together, then we can make this noise or make this big data into knowledge and into discoveries. So lastly, I'm going to introduce my team. We just started last August, so we have lab manager, postdocs, and PhD students from IOB already passed, so there. We have a very interdisciplinary team. People are specialized in mouse studies, cell culture, bioinformatics, every aspect, because aging is so complicated. We need all kinds of talents to come on board and tackle this from different angles and work together. And we also have a postdoc, you know. So he is actively exercising and waiting for us to deliver the next generation of therapeutics. So we're working hard for him. Also, we have coffee to help us achieve that. What's the missing piece? Yeah, we need more people, more talent to come on board and work together to tackle this very complicated problem of aging that affects each and every one of us. So through the years, I've been collaborating with so many amazing labs, collaborators. Everybody have their own special niche to tackle this same problem of aging. So here in UGA, my goal is try to build interdisciplinary team that span from computer science, bench work, and also we have veterinary school here, which is a huge advantage. So we can test the drugs quickly in dog patients, help our best friends. And we also have school medicine. So you will identify any drug candidate, we can move quickly into clinical trials. So UGA is really a perfect place, especially the iOS program. Will allow us to work together as interdisciplinary team. We don't work together individually, but we can work as a team to tackle this complicated problem. So lastly, special thanks to the universe. So make me so humble, like so significant, just like Carson said, no matter stuff. But I know we're tiny, we're small. I think we can dream big. So I would say we also made of dreams. Yeah, we can dream bigger. Also, as I mentioned, the first human TED Talk. After about 10 years, I met him on the aging conference. So I thank him for his hyped TED talk that get me into this field, totally changing my career. Yeah. But recently I just learned that sadly using abstinence file or whatever.
Oh.
But I still appreciate his TED talk. Changing my life. Also the two monkeys.
Okay.
Not two monkeys.
Those are not.
Two group of monkeys, right? So they wouldn't do diet. Behind that, it's a scientist team. So I got to meet this amazing team led by Dr. Anderson. She spent about 30 years doing one experiment. So, and show people this milestone revolutionary work in any field, right? Just to prove people that aging research is real. That changed my career totally. So thanks to all of them. And I think science is motivated by curiosity, but sometimes also motivated fear, right? But fear of death. And this fear motivated me to be curious about how we can do something about this. So along the way, so many people, no matter what kind, they will motivate you. And their talk, their research. So I hope when I start my career here, I can motivate the next generation of scientists. Someday they will change their career to something significant. So that we won't be small. We can still do something significant. So lastly for this group, so how do I take this opportunity to train the next generation of scientists? Right? So we have a lot of research opportunities in the lab. So basically for every project, we are covering from chemistry, biology, drug design, synthesis, and all chemical biology, cellular biology, molecular biology, and machine learning, AI, bioinformatics. No one can drive the project all the way down by themselves. So we need a team. So we have a lot of opportunities for you to be exposed to this interdisciplinary projects. So from the perspective of IOB, we can work together on multi-omics analysis on these data sets from the aging biology, also from the drug studies. And you are interested, we can also do drug design. Can expand your expertise to drug design, or we can do virtual screening as well. But the ultimate goal is to turn data into discoveries. And in my lab, we have regular weekly meetings so people can share their research progress. And we also have one-on-one meetings, unlimited one-on-one meetings. You just knock on my door, discuss projects anytime. We also have subgroup meetings. We have so many diverse projects. We can discuss that in different small groups. I'm not hardcore bioinformatics expert, so if needed, we can find a commenter through IOB or AI Institute to dive deeper into these computational approaches to solve the biology problems together. So what's unique for us? We have very unique in-house data sets for you to play with. And you can't find this anywhere. And we have very complicated models. So the goal is to build this reproducible pipeline, also with wet lab validation. And also I encourage my students to participate in every step of the science. So I will encourage them to go to conferences, to talk about their science, introduce this complicated concept to the general audience. Also, I want to involve them to grant application, so they will know how to write their own grant, apply for their fellowship. And then in the end, I hope everybody, based on their interests, they can learn from each lab members because we are so diverse in the lab. So, so we're recruiting students in almost all fields: computational field, wet lab field, biology or chemistry, everything. So I think you have the curiosity, you have the motivation. Yeah, you are welcome to join us. Yeah, that's it. Any questions?
Actually, that's for a couple questions.
Yeah, sure.
Yeah, so I know that like with caloric restriction boosts autophagy, and that's like a big helps with increasing lifespan, I guess, is killing these senescent cells. Is it like a similar mechanism to autophagy, but just like
Oh, that's a good one, actually. Autophagy is actually behind the mechanism of CR, right? So when you restrict your diet, so your cells will become hungry. They will try to find resource, recycle, like the unhealthy cells or like useless cells in the body. They will turn on autophagy. So that's kind of one of the big mechanism of CR. And currently, that's very hot field. So people are developing autophagy activators to mimic this effect. You don't need to do diet, but you can take pills. Drugs can activate your autophagy, so your cells will have this mechanism to recycle this proteins or cells in your body, so that you can achieve the same effect as the CR, like monkeys. So the two monkeys, the monkeys were doing CR, and you can take autophagy activators and achieve the same. That's kind of cool. We first need to know the biology. How does CR work? You can purchase. What can we do from that? That's kind of great question.
So I know that there are a lot of research on health issues, including aging. Like it sort of focuses more on men, but like women have a lot of very specific things that are different than men when it comes to aging, like going through menopause. And I feel like there's every day, like I learned some like new horrific thing that's gonna happen after menopause that I never heard of in my life. Like now you're gonna have like a frozen shoulder or something. And I was like, what? Like, how does that have to do with menopause? So I was kind of wondering your perspective on that, and if you have any plans to like look specifically at like aging in women.
Yes, that's a very important topic. Actually, this topic is getting more and more attention recently. So there are several groups working on this particularly. So focusing on ovarian aging, right? Because we believe what makes men and the sex difference is the hormonal or the reproductive organs, and women, menopause mostly caused by the reproductive system aging. So now the scientists at which is we collaborate together on this project, look at how ovarian cells, there are zombie cells in the ovarian as well, and also other mechanisms, how we can make it very younger, so we can extend the menopause period, so women can live more healthier, they have more reproductive years. So that's a very important topic. Also, that's a very, I say, significant observation for drug interventions. For drugs, that some of the drugs really work in male only, sometimes work in female only. You can see the obvious sex differences. Yeah, we don't know why, but I would guess it has to relate with the reproductive system or the hormonal system. And speaking of that, like this ESA, I believe may also have sex differences because it rely on the iron levels. Menopause periods can change your iron levels and affect the effect of drugs. So I think this sex difference is very important in aging research. Right? As you can see that women tend to age faster, but they live longer. That's also interesting study in Texas. Let's say probably this related to hormone. Very interesting. Right? So if you capture male mice, they live longer. So I think the cholesterol or this male hormone make people live shorter. So I think that's a very interesting study. Like capture the male mice, and they live longer. And the earlier the better. So but I don't know how that translates to human beings, but this is biology. So very interesting biology. This is, we can tweak this reproductive system or hormonal system. So very interesting.
So if I understood correctly, use the alpha ESA to selectively kill the senescent cells.
Mhm.
But on the other hand, we know, like on the other extreme, if cells achieve immortality, they can grow into cancerous cells. So is there any possibility that this treatment will actually like induce cells to become immortal and like probably go the other way?
Yeah, you should join my lab. This is very, we are working on that actually. We're using the ESA to target cancer, and the ESA works very well to kill cancer cells, because most cancer cells are resistant to apoptosis, like chemotherapeutics. So the ESA is targeting ferroptosis, like induced ferroptosis. So it not only push senescent cell death through ferroptosis, but also cancer cell death through ferroptosis. So we're testing that, and yeah. So great question.
I have a question which is not so much about the science side, but you really cool experiments, really cool research direction, love it. And I see lots of potential for bioinformatics projects there. But you mentioned that you would potentially co-mentor with bioinformatics PIs and stuff. But where is your like core bioinformatics expertise? Like,
oh yeah, that's a great question. So honestly, I just know minimum like bioinformatics. So because you look at all the multi-omics analysis like we did, so it requires minimal coding, especially with AI. So probably you don't need coding. So that's why I just know a little bit of coding. That's good enough for me to do the analysis. But I believe bioinformatics approaches like mathematics, right? Or
computational biology.
yes, that's very important. That's why you will need, let's say, I know the workflow, I know the question we want to ask, right? What problems we want to tackle. And you will need particularly to dive into like particular algorithms or mathematics. In that case, we will find a co-mentor. And honestly, I cannot co-mentor in that aspect. But for the daily analysis of the pipelines, that I can use this.
Are the pipelines use cluster computers and Unix, like, or is it like some other way?
We have the Garak account. We are also building the GitHub. So we will have the whole computational pipeline in the lab, not only bioinformatics side, but also computational drug discovery side as well. So we're trying to integrate these different platforms together. We have the Garak support, and we also have. I'm trying to build a computational sub lab in my lab. So so we're building that pipeline and also the facility.
Thank you.
Yeah.
We have 2 questions from Zoom. You can start with Kashan. Would you like me to repeat or would you ask? All right. Well, Kashan said many PUFAs like DHA and LA. In addition to ESA, are capable of inducing ferroptosis. Why did they show up as senolytics in the screening experiments?
Great question. Yeah, I haven't touched that. So the good fish oils, right? The fish oil that we take every day, like DHA, DPA, you see the structure of them. As I said, they have disrupted conjugation system. Okay, cool. So ESA is very unique and it's totally different with all the other categories of fatty acid because of conjugation system. All the fish oils, they don't kill zombie cells because they have this disrupted CH2 carbon here. So conjugation means head to to tail conjugate. So only these probably 4 fatty acids that we tested have senolytic effect. For this, including all the good fish oils, they don't have any senolytic effect. Also, you don't want fish oil to kill your zombie cells because fish oil usually get into your brain. So that's why also explain why people taking fish oil, they don't have this neurodegeneration problems. So fish oil won't kill their neuron cells. So, but ESA is special because of the conjugated structure. So,
And from Sharif, the question is, with machine learning, are you screening for drugs for a particular pathway or for all the biochemical pathways, molecules, and all of this? Or the pathways that you're getting from GSEA? Correct me if I'm wrong, but I think predicting drug activity from SMILE strings can quickly turn into a more complicated problem to solve with more pathways.
yeah, great question. So we are not, as I said, this is a phenotype-based drug discovery. So we feed machine with drug structures and activity. So there's no target involved, no pathway involved. So that's why this is harder. It's more challenge machine learning pipeline because we are counting on the machine learning to figure out this black box. And why the structure, just because the structure change cause the activity, without involve chemical protein structures or pathways. So that's kind of hard. So that's it, but it works well. With human in the loop, you have to give machine human domain knowledge as feedback. So then tweak this algorithm to get a model. So it's not easy, but it works. But having said that, we can definitely do that. This is because this is phenotype-based drug discovery. You feed target-based drug discovery data. So you have drug structures, also drug target. So in that way, you probably don't necessarily need machine learning. You can just use traditional computational drug design to dock your do virtual screening, like with AlphaFold, or dock your drugs into the protein pocket and do the virtual screening in that way. Of course, you can also, you have the large screening data set, you can also use machine learning to predict that. That'll be easier, less challenging than this phenotype-based approach. Any other things? If you do, please send me email or stop by my office anytime. We have coffee.
Do we need another round? Lars?
Oh, yeah.